First, let's go over the structure of the muscle. Tendons connect the skeletal muscles to the bones, and within the muscle there are many more parts, starting with epimysium. This is connective tissue that covers the muscle and reduces friction between muscle and the surrounding tissue. Then comes perimysium. This is connective tissue within the muscle. Then the fascicle. Fascicles are subfibers of the muscle that contain nerves and capillaries. Next we have endomysium, connective tissue inside of the fascicle. A single muscle cell is known as a myofiber, which is a really long, multinucleated cell. It is multinucleated, so less time has to pass while the information from the DNA is traveling along the muscle tissue. Next we have the sarcomere. This is a long fibrous filament that contains actin and myosin proteins. Now let's go over the sliding filament theory. Essentially, the diagram here is a close-up of the sarcomere. Within the sarcomere, there is the myosin and actin. In order to flex the muscle, the myosin must grab onto the actin and move it. However, in order to keep the muscles from constantly being contracted, the myosin and actin are separated by tropomyosin. In order to move the tropomyosin out of the way, calcium ions come into the sarcoplasmic reticulum and attach to troponin on the tropomyosin, pushing it out of the way. Then, the myosin grabs onto the actin. Finally, let's go over skeletal muscle regulation. First, we have the sarcoplasmic reticulum. This is an organelle that stores calcium ions. Then we have acetylcholine, which is released in the synaptic terminal, which is the end of the nerve cell, across the synaptic cleft that binds the receptor proteins on the muscle fiber's plasma membrane, triggering an actin potential in the muscle fiber. Action potential travels along the plasma membrane and down the T-tubule. Action potential triggers the release of calcium ions from the sarcoplasmic reticulum, which occurs through facilitated diffusion. Then calcium ions bind to troponin, and the actin and the myosin binding sites are exposed. Then, myosin cross-bridges alternately attach to actin and detach, pulling thin filaments towards the center of the sarcomere. ATP powers this process, known as active transport. Then we have cytosolic calcium ions being removed through active transport in the sarcomere after the protein potential ends and the message ends. Cytosolic calcium is removed through active transport because ATP is required to move the calcium against its concentration gradient. Finally, tropomyosin, which blocks myosin binding sites, is restored, and the contraction of the muscle ends, leading the muscle to relax.
What is dystonia and how does it function? Dystonia is a disorder characterized by involuntary muscle contractions that cause slow, repetitive movements or abnormal postures. The movements may be painful, and some individuals with dystonia may have a tremor or other neurological features. There are several different forms of dystonia that may affect only one muscle, groups of muscles, or muscles throughout the body. Some forms of dystonia are genetic, but the cause for the majority of cases is not known. Researchers believe that dystonia results from an abnormality in or damage to the basal ganglia or other brain regions that control movement. There may also be abnormalities in the brain's ability to process neurotransmitters that allow brain cells to communicate with each other.